is actually a possibility. If that question can be answered for me, then that is probably the main reason why I don't accept evolutionary theory as being true.
Okay, so let me try to unpack that. So I heard two distinct questions there, and maybe that's part of the problem. One, you talked about the evolution of major anatomical features. Mm-hmm. And then you tied that with the notion of genus level adaptation. And I always find it interesting because what you said was you accept evolution. You accept evolution, generally speaking. I think you mentioned the notion of ring species and change over time. But you seem to be saying there seems to be some barrier at the genus level. And I'm just trying to be clear on something. Do you understand?
Let me answer. Let me clarify.
There can be anatomical differences among the same genus.
Right. So let me be very specific. Here's the reason. Here's the reason why I said that. There's domains, kingdoms, phylum,
kingdom, phylum,
class, order, family, genus, species. Yeah.
And that's so usually taxonomy. It's become more complex since days of Linnaeus.
That's fine. That's fine. That's not my issue.
Roadmap. Yeah.
That's not my issue. Here's the point. The point is that specifically when people want to talk about macroevolution, they want to talk about change occurring at or above the species level. And rather than getting into a game of semantics, I want to get into specifics as to whether or not an accumulation of changes can actually occur to cause all organisms to diverge and be related to one another. So if I'm looking at something like the different domains of life, eukaryotic cells, prokaryotic cells, and archaeobacteria, or if I'm looking at a dolphin and I'm looking at an elephant, or if I'm looking at an elephant and an ant, there are major anatomical features between these different organisms that need to be accounted for if we're going to say that they are related to one another. So the question isn't whether or not change happens. When we say it in the confines of evolution, it's a very loose term. Change happens, we all agree with that. But the question is, if that change is allowed to continue to happen, whose conclusion of that change is correct? The evolutionist says that if you allow the change to continue to happen, the organism is going to become distinct, producing a variety of different organisms. There's other possibilities on the table that if you allow the changes to continue, the change will continue, but the organisms might inherit some sort of genetic defects, or nothing might happen at all. So there was a variety of conclusions that might be on the table for the change that could possibly happen. The question is that if the evolutionist is going to claim that evolutionary common descent is true, then there's a burden of proof there that the evolutionist needs to meet. And thus far, I've never seen that burden of proof met. So I'm trying to get into specifics. That's the reason why
it's kind of tough because you're kind of all over the map here, biologically speaking, because first you were kind of focusing on the genus level, which I'm cool. That's a good question. Then you mentioned the notion of like elephants and ants. And the distinction between those two groups is far more fundamental than genus. I'm sure you realize I'm not trying to be a pedant here. In fact, the distinction between the elephants and ants goes back to the protostome versus deuterostome, the way that our embryos cleave. One has radial cleavage, the other has symmetrical cleavage. So the fundamental difference between those two groups really goes back to evo-devo and the development of the embryo, beyond just anatomical differences. I mean, it's fundamental to the way we develop as organisms from gestation.
All organisms.
All I'm trying to say here is that when you're talking about these traits, are you talking about the formation of the limb? Are you talking about the cleavage of the embryo? I mean, they're so broad, and they can be so broad, rather. They range very, very highly, obviously. And the level of interrelation can vary tremendously across whatever taxa you're trying to analyze. So I'm not really sure how to answer this or even start this conversation.
Well, I can answer this very, very easily. It's just, the point is that if we stay at the description that typical evolutionists go at, which is variety, and we're just talking about change, speciation, events, that doesn't answer the question. But if we're going to be talking about these major anatomical features, they all come down to one particular problem: whether or not the genetic changes can accumulate to build or to be translated to build
manifest,
these. So it all comes down to manifest. That's the word that I'm looking for.
It's a good word, yeah.
So I mean, I can get into specifics if you would like me to.
Oh, sure. Well, but I can help you there. So essentially, there's a problem I see. But to answer your question directly, my work actually at the graduate level, I mentioned was on parasitoid diversity. And so basically these are very small wasps. And these are parasitic wasps that attack a species of fly. It's a midge fly. And so what I discovered is that the diversity of these wasps was very irregular because we had the same, we call them morphospecies. They looked the same. You couldn't tell them apart. Analyzing, I spent hours looking at them under microscopes. They're only about 3.5 millimeters long. And morphologically, they were indistinct. You couldn't tell which was which. And yet, genetically, when we sequenced like the 35S RNA gene, which is a standard barcoding gene, we found significant divergence, such that we had essentially two separate breeding populations. And that was interesting because the midge fly population, their prey, they would lay their eggs on the larvae, and their developing larvae would eat the insides of the fly larvae, and that's how they lived. That was their life cycle. Even though they had the same thing, they were the same morphospecies, genetically they were 4 distinct species. And so in this case, we're talking about genetics and saying that these flies, despite the fact they live on the same plant, goldenrod, they form the same type of gall, how they live, basically where they develop as larvae, and their parasitoids look the same as far as we can tell. Genetically speaking, they're not crossbreeding. They're becoming isolated, and they're accumulating mutations at whatever rate they do. There was some microsatellite work. I don't want to bore you with details, but we did tests on this system to kind of get some idea of what's going on here. And even though no significant morpho changes were happening, their anatomy wasn't changing, we were able to see that their genes were changing, and they were not freely interbreeding as we would expect. So you can't have that.
You missed the question.
Change. Well, but that's
the point.
Well, that's not the point. That's the first part, which is to say, why would you expect evolution to manifest some particular change? You obviously wouldn't. But to your point, your question was why?
You're not answering my question.
I'm not answering the question at all, man. If I may. So the problem here is you're saying, well, why is there some limitation? You know, if we're talking about just the diversity among genetics, what makes you think that the small changes couldn't accumulate?
Oh, come on.
Okay. So I know you want to get on this too, Veckel, because Veckel's been here the whole time, and Veckel comes into my rooms a lot, and he sees the same problem. We actually just tackled this, and you did what I said.
I apologize.
Yeah, well, I know, but you did what I was complaining evolutionists tend to do. I don't have a problem with whether or not you're going to see two species not breeding with one another. I don't have a problem with that. And you don't answer the question by saying, well, I don't see why it, whatever, or can't ponder some sort of other question.
Well, aren't you asking about some barrier? Have you established this barrier? I mean, why do you think it couldn't accumulate? Like, what problem should I overcome here to address the questions? What I'm asking.
The question is whether or not the accumulation of changes have the creative ability to build the anatomical features that we see across with these different organisms.
Yeah.
So I said, let's get into specifics here, right?
Sure.
I'm somebody who tries to get into specifics. I'm somebody who supports the idea of intelligent design. If you put intelligent design and evolutionary theory both on the table to see what best accounts for what we see inside of nature. Inside of nature, you see systems in life that we know only come from an intelligent creator. And I know you guys already know what I'm going to get at, the people who are watching. Genetics itself, that's an obvious given. I mean, that's probably one of the most powerful arguments for intelligent design. And of course, an evolutionist will say, well, of course, because there's genetic homologies there, then you can compare that with the fossil record, and it's evidence for evolution as well. So we can have a tit for tat there as well. But you move into specific categories where you see certain objects inside of nature that can't be accounted for evolutionary theory, like the ATP synthase, right? It's a turbine. It is what it is. It's a turbine. There is no other system out there available that can account that evolutionary theory can utilize to say, well, therefore, evolutionary theory is true. Do you get what I'm trying to say? They got like the hexameric helicases, and they can compare their homologies and their proteins and assume, well, one came from the other or diverged from the other. But comparing homologies doesn't show us whether or not the mechanisms of evolution can account for this change. So going back to my original question that I was asking, the question is whether or not there has been tested mechanisms in the laboratory to show that these changes are actually a possibility. That's what I'm interested in.
Yes.
We have an explanation here that we know intelligent agents have always built turbines. We know that for a fact, but we don't know whether or not evolutionary theory can produce the same exact thing.
No, I understand. I appreciate your clarification. So I do object because, first off, comparing the hypothesis, to be generous, of intelligent design to the theory of evolution is simply incomparable. There's no connection whatsoever to the explanatory scope or framework, the level of rigor, or even the experimental support. I mean, to even compare those two is ludicrous. Intelligent design is at best probably a creationist idea that could be tested. Whether or not it could develop beyond that, I leave that to the proponents. I mean, famously, Michael just wrote a new book that got panned in Nature, and he's complaining about it. But essentially, he's not changed his argument. And I think what's so sad about it is the idea that these could be tested. These could be ideas that could be developed. And yet people like William Dembski or Michael Behe or Stephen Meyer, they're not really taking a scientific route to refine and try to convince their peers in their respective scientific communities of their ideas. So you say things like, well, we know these things only are created or are only manifested by intelligence. I don't think we know that. I mean, what precisely are you saying? I mean, say, for instance, in the case of ATP synthase, ATP synthase is not a turbine. It's a protein. Are you aware of that? Like, there's a difference between something you might use, like, you know, you could say that the ion pump in the stomach is like a outboard motor or like a sump pump, but it's not literally a pump. It's a protein. And you say that because it's a clustering of chemicals that are manufactured in a way that is in no way, shape, or form analogous to what humans do, or what humans do with their inventions or creations, precisely because they exist in self-replicating systems. And so when you're talking about the notion of, say, you mentioned also the genome, you know, DNA. I'm sorry, no, I'm not sure if you know much about the nature of how DNA works, but it is a mess. You know, we have something like 3 billion coding base pairs in our genome, and only a very small fraction actually code for the output. On top of that, you know, quite a bit more does regulatory action or is associated with proteins. But there's a lot of junk in there. There's a lot of stuff that does really nothing, including repeats, tandem repeats, CPG islands, and even things at the end of our chromosomes like telomeres. So it's really a Rube Goldberg contraction, and whether or not you think that's believable or something that could result from accumulations of evolution, the point is that it's at least possible in the sense that these are self-replicating systems that exist independently. And unlike turbines, they cannot self-replicate.
Okay, so are you finished with your response?
Well, yeah, sure, for that, yeah.
Well, what you just said is 100% incorrect.
How so? So can turbines self-replicate?
The ATP synthase is...
No, wait, can turbines self-replicate?
Hold on a second. Don't start over-talking. Okay? The ATP synthase is a turbine that is located inside of the mitochondria, which is part of a system, obviously, the basic unit of life that does self-replicate. You're right. But the ATP synthase has multiple subunits in the C ring, which is where the rotor would be. There, the shaft that would go down to the catalytic core opens up so that the additional phosphate can be added. It is in every dimension the exact same as many other turbines. What is a turbine? A turbine is a utilization system. You got steam turbines, Pelton turbines, where they might use some sort of mechanical energy to produce some sort of electrical energy, and they might use water or wind to produce this energy. It's the exact same thing with the ATP synthase. It's got the same parts and it's got the same overall purpose. It is a turbine. Every single scholarly article that I've ever read regarding this topic in mainstream journals describes it and explains it exactly like this. So I don't know why you would say that it isn't a turbine. That's just, in regards to the literature, that's just, it's factually.
Well, wait a minute.
So what do you say? Hold on a second. Just let me answer all of your points that you just made right now.
Okay.
You were saying a lot of things that were incorrect right here. The ATP, you're right about, there is a mess, or I wouldn't say it's a mess. I wouldn't word it the way that you word it, but there is a sort of randomness that happens with with DNA, but it's a 3 out of 4 error correcting code as well. And the reason why it's analogized to being information is because of the specific sequences of those characters down an information chain. So whether there's the way that you put it, whether it's a mess, it's irrelevant to the point as to whether or not it can be inferenced, or you can use some sort of abductive reasoning to come to a conclusion that an intelligence was behind it. There might be some mess behind it as well on the side, but if its properties can be connected to that of an intelligent agent, then the inference is reasonable to make. So, I mean, that point in itself was irrelevant. So when you say, well, does the ATP do turbines self-replicate? Well, what self-replicates is coded information that we know only comes from an intelligent agent that we're observing inside of the cell. That's the point here. So without you having that information to self-replicate, you're not going to get the ATP synthase, which is a turbine, to self-replicate. So the information that's needed behind all of this to begin with is the inference or the indicator of an intelligent agent. So what you said, it was all jumbled up.
No, so there's some problems here because first off, you're clear in saying I wasn't 100% incorrect. So I'm just happy that you're a little hyperbolic, which is fine. The problem with this is I'm sorry, no, that this is not what the scientific literature actually says about these. I posted 2 links to scientific journal articles which describe the evolution of this. So a very simple search of literature, even by a novice, could see that there are a number of explanations, including tests that seek to confirm the idea that these are evolved structures. When we're talking about the notion of it's actually a rotor, or it's actually a turbine, no, that's an analogy. It's like saying that the flagellum, bacterial flagellum, is an outboard motor. You know, these are proteins, these are chemicals, these are structures. And I have to disagree with you that the connection to the genetic code is not irrelevant, precisely because, as you implied, the coding for the continued construction and propagation of these structures inside the cell is in the code. The code is there. And in fact, the structures that produce these things, that assemble them, the chaperonins, the ribosomes, the lipases, or the Golgi apparatus, the internal structures are likewise coded within the genome. So it's all connected in that sense because you have a self-replicating system, the cell, which contains chemicals we call DNA, that outputs other chemicals which we call proteins and of that sort. And when you say the word code and connected to information, I think that's the fallacy that's beneath the intelligent design argument. Because as William Dembski would talk about specified complex information, if you're trying to conclude that some set of information is the result of design, as is often said, you need to have some metric to distinguish that from undesigned information. And so far, the intelligent design community has rejected the calls to have rigorous definitions of SPI, or specified complex information. And it's almost like a buzzword with no real meaning. Like, what do you mean by information? If I gave you a set of information, how would you tell one set from another with regards to its origin being design?
Okay, so you finished? I just gave you one article right there. I can look for many, many more that describe ATP synthase as, this one specifically, speaking of the FATPase, being a turbine. Any article that you're going to give me in regard to the evolution of the ATP synthase is going to only have to do with comparing the homologies of the proteins involved. That's why I already mentioned the hexameric helicases earlier on in this hangout. So you have no article at all. There is no article that exists anywhere in the literature that can show the evolution of the ATP synthase. What you can do is show already existing features and then use your imagination to connect the dots by looking at homologies. That's not the evidence that I'm looking for. What I'm looking for is whether or not there have been tests in the laboratory to show that the mechanisms of evolution can account for these distinctive changes. Instead, all we're getting is, well, here we already have an existing system. We have another existing system over here that looks similar to this one. Therefore, we will assume common ancestry. That's not answering my question. If there is evidence for this, then we need to see what tests have there been done in the laboratory to show that these changes can occur. And thus far, the only known fact that we know that can account for a turbine, which I've given you an article describing the ATP synthase as exactly that, is an intelligent agent. That is the only known cause that we know of that can produce a turbine. That's not an opinion, that's a fact.
Well, no, I'm sorry. That's simply not correct, because even so, as I said before, this is the analogy that's used to describe the action. It's like saying that DNA is a code. DNA is not a code. DNA is a process of chemicals. And we can say, well, we can look at it and talk about the genetic code, talk about how the chemicals interact in a way that makes sense to us. But in the sense of a code being a message to be transmitted from one thinking being to another, the analogy doesn't quite hold. And I think that therein lies the problem, because if you're going to say, well, let's call it the DNA synthase, or ATP synthase rather, let's call it a turbine. I'm okay with that analogy. If you want to use that literally, more literally than I would have, that's okay too. In that case, you have a problem, because now we have the example of turbines that don't come about by intelligence, because we have them in living cells. They're manufactured by cells, like without the direction of teleological agency, in the sense of, can a bacteria have teleology rather. So the question becomes, when you're talking about this and saying, well, can evolution do this? Are you kind of saying yes? Because when we're proposing these issues, when we say, where did this come from? The reliance on homologies is to say, hey, these exist. Of course, ATP is made up of subunits, naturally. These structures exist in other forms, in other parts of nature. We can actually track their development throughout different systems and evolutionary lineages. When we say this, we say, well, are you saying now, this is a fine line, that say the ATP synthase in, say, mammals, is that the same one that existed in bacteria? Did the designer make those two separately? Or could we say potentially, could there be a designer, say, of the bacterial ATP synthase, the first one, and evolution takes it the rest of the way? And so the issue becomes, well, where are you drawing the line with regards to what evolution can and can't do? Because I would, going back to the issue of limbs too, if you're saying, can evolution modify already existing structure, the answer seems to be yes. Whether it be the case of the blood clotting cascade, the evolution of anti-freezing compounds in Arctic fish, or say the evolution of bird limbs from theropod dinosaurs. I mean, there's very clear evidence genetically, fossily, and morphologically to suggest that these structures can be modified. So at best, are you saying that the current conception or the current manifestation of ATP synthase could have been evolved from a more basal ATP synthase?
Well, there's a lot to unpack with what you just said.
Yeah,
I wouldn't disagree with you that deformities can occur and that changes in already existing features can occur. But whether or not the accumulation of changes as regards to evolutionary theory can accumulate and build a distinctive feature, that's something that evolutionary theory has never been able to account for. So when I'm looking at homology between two already existing features that have functionality behind them, to assume that one diverge from another, it's speculation. I can come to that conclusion if I already have preconceived notions as an evolutionist, but that exists in my head, and it's not the scientific method. The scientific method utilizes tests and experiments, and of course mechanisms. You have to have some sort of driving force to get somewhere in order to utilize some sort of facts and come to a conclusion. But when you say analogy and you're trying to jump on this word, this description of analogy, you're misunderstanding. When you're looking at the ATP synthase, it's not just analogous. It is what it is. It is factually a turbine. In all dimension, it is factually a turbine. But when you're looking at, for example, the brain is typically analogized to a computer system. But because the way that the astrocytes work with the neurons and the way that it processes information, you get the stimuli through your skin and the information is sent back and forth through your central nervous system. It's analogized to a computer because they're very similar, but they're not the same thing because the brain doesn't operate on the motherboard, CPU, and so on. It doesn't have the same parts, but It's comparable. You can analogize it. With the ATP synthase, you don't just analogize it. It is the same structure. What you see inside of the basic unit of life is the same thing that you see outside of life. The only difference is the material that's used and the fact that it's on a smaller scale. That's the only difference there. But even if we were to grant you that and just use the description of it being analogy, like going to the brain, the fact that we can only analogize the systems that we're seeing inside of life to that of our own created systems and not that of some unintended gradual process is still an indicator of a design which gives us more argumentative power than the evolutionist does, because there is no real world open demonstration of an accumulation of changes building these distinctive features. Right? So we can argue over all sorts of things vaguely all day long. I mean, and we would probably both agree on it. Right, evolution can cause changes to happen, even possibly new traits, like the ability to digest myelinase, or there's all sorts of different mutations that have allowed new traits. But when you get to specifics of the anatomical features that we know for a fact, based on the cause and effect, have only come from a creator, like a turbine, there is no accountability. Evolution has no open real-world demonstration to account for these things. So,
well, I'm sorry, I have to correct you once more. I mean, you keep on saying only a creator, and that has not been demonstrated. At best, you're saying we have examples of creators, like of humans, basically, designing turbines with teleological intent. And so my analogy I always like to use about this is of course I've done before, so it's somewhat canned, is imagine, if you will, that I'm hiring contractors to build a lake behind my house and stock it with fish, so I can observe them. I design the lake, they come over, the landscapers, dig the lake, line it, fill it with water, stock it with fish, whole 9 yards, right? I invite you over and I say, hey, let's go out and get some grasshoppers, or dig up some night crawlers to go fishing in my new lake. And so we walk into the woods, and lo and behold, we come across a lake. And I say, oh, look, there's a lake here. Well, this must have been built by landscapers. So this must be an invention. Someone made this.
Sorry, I'm sorry. You cut out for me there. Can you repeat that, please?
No, so I'd say, then that must mean, we come across the lake in the woods, and I tell you, someone must have built this lake. Like, I can tell because I have a landscaped lake behind my house. I mean, I saw it happen. I took pictures. I can prove it to you. That lake was designed. This lake here must be the result of a creator. Now, would you say that was a faulty reasoning on my part? Would you say, well, hey, no, actually, maybe this lake is natural because natural processes can form lakes?
Well, I would say that the difference would be is that there is only one explanation on the table for a designer building turbines, right? We only know that an intelligent designer builds turbines. That's the point here. Well, if we have two options on the table, like man-made lakes versus naturally occurring lakes, then we have two options on the table. So it would be unreasonable for me to say, well, this was for a fact. There is no other explanation for this.
So first off, do I have to point out that's literally argument from ignorance fallacy? That you're saying, well, we only have this answer, so it must be it?
No, that's not a misunderstanding. No.
How is that not the definition of the argument from ignorance?
No. See, that's not how it works, though. Science doesn't say, no, science doesn't work like that. You don't say, well, all of the evidence points in this direction, but it could possibly be another explanation. So you don't get to hold to this only known explanation for an alternative. See, that's the way that you're approaching it. I'm saying, since the only known place that we know of thus far is an intelligent creator has produced turbines and nowhere else, then I'm going to hold to the facts that are on the table rather than somebody's alternative that they're assuming by faith.
No, okay, so first off, I'm simply going to ignore a lot of the implications of that last statement and just saunter on with the conversation. And what we're dealing with here is I've tried to point out that your reasoning on that point is flawed. And it's flawed in a very specific way because it's called the argument of ignorance fallacy. You're trying to draw a conclusion by saying we only have this answer.
It's the scientific method.
Well, wait a minute. Well, actually, it's not the scientific method. But the point I'm trying to make here for the positive case is to say, I'm sorry, no. Not only have I given you scientific articles, which you're free to check at your leisure, describing hypotheses that have been tested to some degree of evolutionary pathways of these structures. But specifically, the question you asked was, can we demonstrate that evolution has this power? Now, I'll be free to say that we do not have a mechanistic, step-by-step explanation of the development and evolution of ATP synthase. But the question you asked, which I think is a very good question, was, well, can evolution do this? Is it reasonable to propose evolution as an alternative? And in the case of the last article I posted about the evolution of anti-freezing proteins in fish, we can see that, yes, it does have this power. It's not about, well, there can be mutations, there can be deformities, as you use the word. No, unrelated sections of genes can become deactivated, can become pseudogenes, can even randomly mutate, become reactivated, reformed in different structures, whether it's the varied origin of the formerly favored example of this, the bacterial flagellum, whether it's the anti-freezing proteins in Arctic fish. In some cases, we do have, in the case of, say, the Arctic fish, a step-by-step pathway of how this protein evolved and changed into what it currently does, how it's called exaptation, how one function can be co-opted into another, in some cases unrelated function, through a stepwise change pathway.
Well, let's be very specific.
Attempt to address what you're saying. Is it in the direction of what you're looking for?
Well, let's be very specific here. When we're talking about the ATP synthase, the enzyme that is the ATP synthase, you're dealing with 10 to 15 different protein subunits just in the C ring. In the catalytic core, you got 3 alpha, 3 beta, then you have gamma, delta, epsilon, multiple different enzyme subunits just located there. Then you got to deal with multiple different protein subunits that are embedded along that mitochondrial matrix. Cytochrome BC1, cytochrome oxidase, and this whole process of glycolysis reaction before that. So when we're talking about the ATP synthase, you're not just dealing with the protein, you're dealing with multiple different proteins that have their complexity and their purposes here, all to do the same job that we're seeing in other turbines outside of the system of life. Pelton turbines, steam turbines, to produce some sort of energy for some system. So I think the issue is a little bit more complex here. But like I said earlier on in this hangout, that we can get into vagaries all day long. I mean, we can talk about all sorts of traits that we would both agree on. Came from evolutionary theory.
Step by step.
It doesn't answer the question, because if there are specific objects that we're finding inside of nature that are needed for life, like the ATP synthase, and that the only explanation that we have thus far is an intelligent agent, then the question isn't being answered, I guess. So,
well, again, I'm sorry. No matter how many times you say that, it's still inaccurate. Because
what is inaccurate?
So the point I'm trying to make here on that is to say, you simply saying that doesn't make the case. I don't think that's an explanation to say that, well, some designer made it. Because that simply pushes the question one more step. Like, who designed the designer, et cetera, et cetera.
Whoa, hold on. That's a separate conversation. You don't need explanation.
Well, but wait a minute.
You're misunderstanding.
But when you're talking about this, you keep on saying, there's no other explanation, and I'm giving you one. And I tried to, it's not a vagary when you ask the question, well, can evolution take previously existing structures or proteins and reassemble them, even in complex assemblages, in a step-by-step pathway?
You're doing two things, though. You're shifting the goalpost, though.
Well, wait a minute, and the answer is yes. And I'm trying to say, in some cases, we have actually found compelling evidence this is the case. In some cases, we've actually found evidence of the step-by-step.
You haven't, though. So,
well, I'm sorry, sir. Yes, we have.
But you haven't. Okay, so I'm going to respond to you now, okay? The first question is that if we were to go by your logic, then all science would go out the window, because all questions that are usually in science, whenever there's a question that's answered, usually that leads to additional questions. Just because we can ask the question, well, who designed the designer? And we might have additional questions after the first question is answered, doesn't mean the first question is therefore invalid. So we can detect intelligent causation by looking at known effects that had been the end result of the cause, which was intelligence. So if we find turbines, we know for a fact, based on our uniform experience, that has come from an intelligent agent. In every instance in the past, as far as our knowledge goes, we might ask, okay, this is what the evidence is showing, that there was a designer. We can detect intelligent causation. But who is the designer of this designer? Who designed that designer? We might have those questions. Those questions may never be answered, but that doesn't make the question go away at all. So in the same way, when you say that, well, we have we have these different explanations, and there are all these explanations that we have out there that can account for these systems. You're not answering the question. Moving on to these, moving on away from the ATP synthase and talking about other things that are unrelated to a specific cause that we know come from an intelligent agent doesn't answer the question and convince me that an intelligent or that your position is correct. So I mean, you changed the topic.
Well, I'm sorry. I was trying to address the question you raised, which was, do we have any experimental results or evidence to suggest that evolution has this power? And if we're trying to say, so the answer to your question is evolution. That's the alternative. And you ask quite rightly, well, is that a valid explanation? Do we have reason to believe that evolution can do this? And you keep on saying this, and what I'm trying to say is it's incorrect. Because when you say, well, we only have one explanation, design or intelligence.
Well, you have a positive.
Well, the answer is no, because if you're going to call ATP synthase a turbine, which is an analogy, I'm okay going with that. That's like saying that DNA is a code. And if you say, well, all codes come from intelligences, I can't grant that, the sense of the first premise, because then your work is done for you. And there's no argument.
Well, hold on a second.
All we have evidence of is a code that exists apart from intelligence. Like with the rotor.
Okay, well, the first thing is that you have an explanation that you're positing, but the explanation that you're positing isn't answering my question. Right? You can posit all day long by giving me this article in an expected or anticipated way how the ATP synthase evolved. But if in that article all they're giving me is what can be conceived in the mind of the people who are talking about homologies, that's not answering my question. I can look at a skateboard and I can look at a motorcycle and look at their comparative parts and assume that one diverged from a common part through random processes a long time ago. It doesn't answer my question. Looking at comparative homologies and looking at comparative parts, it doesn't answer my question. And so
can I try a different way of answering then?
Go ahead. But so, I do. It's getting late, and I do got to get up early.
So the problem I have here is the first objection I have is the notion of the argument from ignorance, the idea that at best you're putting forth one explanation excluding others. And I'm saying there are other explanations. You can say, well, do they have sufficient explanatory power and scope? What I'm attempting to address with the example of, say, the evolution, as you mentioned, nyelinase, good example, the evolution within E. coli of a particular, I think it was a citrate digestion promoter, or in the case of the freezing proteins in Arctic fish, I'm attempting to point to evidence that evolution does have this explanatory power. When you're talking about the notion of, well, what about homologies here? Well, we're talking about homologies that in the DNA, the genetic code, have correlations to the development of the actual protein, the phenotype, so to speak. So it's like saying, well, if I type out, a paragraph, and I give it to you to type out, and you type out a paragraph, and you misstep a letter, and you have a typo, and you give it to someone else, and they copy it themselves, they type it out, they make another mistake at a different location, and this goes on for maybe 5 or 6 different times. Could you organize that series of letters by the original with no typos, and get an idea of in what order did these particular typos accumulate? Because you'd have, in every copy, you'd have the previous errors, but you also have the new errors. Yeah. And that's essentially what we're pointing to. We're saying evolution has some explanatory power. We can demonstrate that it can make changes.
Why haven't you?
We say, what's the origin of the ATP synthase? And we say, aha, there seems to be these homologies throughout evolutionary heritage. And we say, not the things we can't go back in time and see, like, what's the ATP synthase in a protozoan 1 billion years ago? We can say, what does it look like today in the modern evolved ancestors? And we say, it seems like there's a step-by-step pattern where we have these typos that are prevalent, and say this is indicating of a story that is essentially in line with evolutionary expectations.
Okay, so if the topic is specifically trying to narrow down the complexity of anatomical features occurring above the genus, or even within it, if there's a specific topic that's relevant, and you're going on about proteins, that's, like I said, you're going into vagaries. You're not.
I'm sorry, sir. You raised.
Hold on, Velo, hold on. Okay, well then why don't you answer my question regarding?
Well, I'm sorry, I'm really confused.
Because you keep on saying that you do have an alternative explanation, but I don't care about explanations that I got to use my imagination and connect dots for. I'm concerned with explanations that have scientific research behind it to show that it's the case. If I'm looking at homologies and I'm not looking at any tested mechanisms to show the end result, you're not answering my question.
So, first off, I prefer to use the example of, say, the evolution of the avian forearm, and as it relates to theropod dinosaurs, I think that's perhaps one of the best explanations with regards to how these anatomical differences can affect. I would be the first to say that ATP synthase is not an anatomical difference, and it's certainly shared far beyond the genus level. In the case of homologies between animals and in prokaryota. So it's a really bizarre example that you raised, and it's a molecular argument. And again,
who cares? It doesn't matter, though.
Well, I think it does matter.
It doesn't.
Precisely because there are rigorous tested methodologies which I've put forth to you.
Well, the first thing that's important for us to note, though, is that we can, even if we talk about theropod dinosaurs or anything like that, I mean, obviously the answer is pretty obvious there. Anything that we're seeing, especially millions of years ago, we don't see that evolving before our eyes, right? We don't see that happening. So what we would need is some sort of changes or tests in the laboratory that have been experimented on, so that then we can come to an inference on to say, okay, well, this change can occur if we allow it to, what we're observing inside of the fossil record is actually a possibility based on these experiments we have seen and observed inside of the laboratory. We don't have any of that. But dealing with the ATP synthase, it's kind of convenient, right? Because I'm an ID proponent. I support intelligent design. This is what I think is the best explanation regarding to what we're seeing inside of life. So when we get into specifics of intelligent design doesn't say all things in nature were designed. Everything wasn't designed. Intelligent design says that certain patterns in nature are best explained by an intelligent cause because those causes have been observed coming from an intelligent agent. That's what intelligent design says. So if we're looking at specific patterns inside of nature, like I brought up, the DNA code, and you're looking at the ATP synthase, or I would even go as far as to argue maybe the brain itself and its complexity, that these are specific patterns that an evolutionist would have to deal with. When you move on to other topics, which I don't think you can give an answer to either, I think you can try to make an argument, but not deal with the question of whether or not the anatomical features observed have actually been tested in the laboratory to, the mechanisms have been tested to produce those results. I don't think you can give an answer to that. But it's kind of convenient to move away from the ATP synthase, because to me, I get on this topic because it's the most powerful argument. See, when we go on to DNA, the topic can become very confusing. You can get into information theory, Shannon information, where you reduce uncertainty and look at the probabilistic outcome of the characters over time. But it wouldn't be the same thing, specific sequences of information. The topic
wait a minute, and that's the problem.
Hold on. Just really quick. So the point is that with the ATP synthase itself, I just find it very convenient to just try to brush it aside and say, well, you know what, we don't really have an explanation.
I'm sorry, I've given you scientific papers that detail the evolution of ATP synthase.
You haven't. Now I'll look through right now.
And
I see what you're giving me.
If all you're saying is there's no step-by-step mechanistic pathway for the evolution of the structure, I'd say yes, we don't have all the answers. But simply to say, well, maybe a designer did it, I don't think that's a valid answer, especially when I think your rationale is to say, well, we designed these other structures, they're somewhat analogous, therefore something must design this too. And that's
You've given me Wikipedia. You've given me a Wikipedia article.
Have you not seen the cross journals and the NCBI references?
I'm looking at this. I'm looking at all of them. And all of these articles, they're going to be talking about homology.
Just to be clear, you haven't read them, you're saying. You're just looking at them.
The one right now that I'm looking at, I couldn't. You just overtalked me. I couldn't hear what you said.
You're not familiar with them off the bat. You're just saying you suppose they say this.
I've read quite a few articles over the ATP synthase.
But you haven't read these, what you're saying.
I'm clicking into this one right now. So give me
that's right. And by all means, take your time and read them for the future. And it's more than Wikipedia. Although Wikipedia does have some great citations. I highly recommend you check those out. Don't just read the article, read the citations in the article. But the problem I'm broaching here is, one, you've talked about, what does evolution have this power? Does it have the explanatory power?
What was the article that you're referring to, though, that shows the evolution of the ATP synthase?
So there's that issue to say that we can demonstrate that evolution can modify proteins over time. In the case of, say, antifreeze proteins in Arctic fish. So I'd say there's reason, and there's, of course, many examples of this. You can point to the fact that the vast majority, over 99% of the biologists in the field, especially the ones particularly in the biochemical evolution of these structures, disagree with ID proponents. And it's not considered a valid scientific theory, and it doesn't seek to publish its ideas in scientific journals. I think it could become a better theory with regards to how science proposes, a better hypothesis. But as you said, when you talk about specified complex information or information, I don't see how you're identifying as such. Because I keep going back to my objection, which I haven't heard you address. If you could try to address this more directly. At best, you're saying, well, yes, we have examples of designed windmills or rotors and undesigned rotors.
So this one says, motor and molecular motors have directionally along a DNA strand powered by nucleotide hydrolysis. These motors are multimeric ATPases. I'm going to put my glasses on. Give me 1 second.
That's fine. But then the issue
with more than one hydrolysis site, we present here a model for how these motors generate.
Isn't that the problem, though, is that at best you're pointing to an example of an undesigned motor?
This article that you're giving me, it doesn't say anything about it evolving even.
Well, wait a minute.
This is describing its functionality.
Well, that's what I'm saying here, because if you're saying we only have examples of this.
Hold on. I got to stop you.
Please.
You just told me that you gave me articles showing the evolution of the ATP synthase.
Have you checked the PLOS, the Public Library of Online Science article at 11:38, the first thing I posted?
First thing.
Titled Evolution of the F0, F1 ATP synthase in light of the patchy distribution of different bioenergetic pathways across prokaryotes.
I reposted because there's a lot of...
I know it's a lot of words, right? So, but what I'm saying here is apart from whether or not you're willing to accept the literature...
I reposted because I read a lot. There's a lot of links here.
Yeah, 11:38 p.m. It's the only article that links to the journals.plos.org. It's the very first link I posted. After the Common Descent post, sorry.
Journalplos.org?
Yeah, it's the Public Library of Science. So specifically, though, what I'm saying here, and what I haven't heard you address directly, if you could address it more directly, is the idea that at best what you're saying then is that we have an example of an undesigned rotor, because in the case of, say, a bacterial ATP synthase, it's being assembled and constructed specifically from unintelligent causes. In this case, the molecular machinery of the cell. And so whether or not you think that was designed, that's a separate conversation, because the output of this is in fact from a self-replicating system. And that's the difference, because unlike how we as humans build rotors, build turbines, that's not a self-replicating process. It doesn't exist in a process that self-replicates, unlike the cell, correct?
Well, you've been talking a lot while I've been looking at this, and I'd like for us to look at this supposed evidence of the ATP synthase together so we can dissect it for the audience.
Well, instead of the article now, can you address the question I asked you? I'll be happy to repeat it if you missed it.
Well, I can do both. You go ahead and ask me the question again.
Sure. So the issue I'm raising here is then, aren't you simply pointing to the fact that in the case of, say, turbines or rotors, we have examples of designed turbines and rotors. We have examples of undesigned turbines and rotors in the case of, say, the ATP synthase. Because at, say, the bacterial level, they are being assembled and operated by unteleological agency, the self-replicating mechanisms, the molecular biochemical pathways within these cells, correct?
So you're saying that because there's a system in place, and therefore that system in place, therefore makes it invalid?
That's certainly a separate argument you could make about
what you're saying.
What's designed. But I'm saying if you're just talking about the rotor, the turbine, that is in fact being constructed and assembled apart from teleological agency, correct? When the cell assembles an ATP synthase, you're not saying a bacterial cell is intelligent, correct?
I'd say that the cell itself is an indicator of an intelligent agent because it consists of systems that we know have only come from an intelligent agent.
But no, of course not, because if the cell is not intelligent itself and it's producing.
Wait, what?
If the cell, like the question was, and that's not quite the answer to my question.
What do you mean by intelligent itself?
Well, that's a good question. That'd be for you to answer, because if we're talking about,
you asked the question. You asked the question. It doesn't make sense.
Maybe it doesn't make sense. So if we're talking about a bacterial cell, you're not saying the bacterial cell is intelligent, obviously.
What I'm saying, the intelligent design argument specifically goes like this. Certain patterns in nature are best explained by an intelligent cause because those patterns that we see have been observed coming from an intelligent agent. It's not to say that thing. I don't know what you mean by intelligent. It's just to say that there are patterns that we see.
That goes back to my analogy that
if you see a computer, for example, it has random access memory, a CPU, a motherboard. It has plastics and certain materials.
Yes.
And it's got all of those components have been placed together in a coordinated fashion to give an overall function, which is a computer. That experience of a computer, because you are an intelligent agent, you understand, you know for a fact, based on your uniform experience, that system has come from an intelligent agent. It's not to say that the system is intelligent, but the system that you're looking at, the computer, has functionality and patterns, a build quality that you know from experience based on cause and effect has come from an intelligent agent. So with the ATP synthase, because of its properties and because of its overall purpose and functionality, we indicate that it was the end result of intelligent agents because of cause and effect. Similar to evolutionary theory uses an inference of a best explanation. It utilizes certain facts to try to come to a conclusion. That's exactly what intelligent design is doing. Based on our facts that we know from the past, an intelligent agent is the only known cause available thus far to account for this particular system.
And so I think, again, simply restating the position is...
But you said, you've been claiming, I'm not trying to cut you off. I'm not trying to cut you off, but you've been claiming that this article provides the evidence for evolutionary theory in the sense to where...
Well, discusses the evolution of this of the case of the ATP synthase.
So just for clarification, it doesn't deal with just homologies.
So this is the problem here, because when you say it provides the best argument, you're not actually making that argument.
Well, you want to read it together? Let's read it together.
Wait a minute here. So you're making what I would call a logical fallacy. Precisely, and I pointed this out in the case of my pond analogy, because if I'd never seen a naturally occurring pond, and my only experience, say, growing up in the city, was with landscaped ponds, I might be tempted to conclude that erroneous conclusion. And simply saying, well, we haven't seen some other explanation, therefore this explanation must be the best explanation, or even the only explanation, I think that's problematic, especially when there is another explanation that's held to be valid, at least by the vast majority of professionals in the field that actually study this at the academic level. So if you're trying to address that, well, likewise...
The vast majority of the scientists in the field, with all due respect, you're mistaken there. The vast majority of the scientists...
I'm not mistaken, though. I think the problem remains, which is to say...
The problem is that the vast majority of the scientists in the field all acknowledge that the ATP synthase is a turbine.
But they believe...
The vast majority of the scientists, even, for example, you disagreed with the analogy of DNA being a code.
Well, it's an analogy.
It's comparison to that of our own creation.
Yeah,
even Richard Dawkins has acknowledged that it is, in every single way, he described it exactly how ID proponents describe it.
This is the problem.
Even Craig Venter describes it exactly how ID proponents acknowledge it. So here's just really quick, because this is really important. The only difference between intelligent design proponents and mainstream science is that they take the evidence to its conclusion. All the scientists on both sides of the fence acknowledge the data for what it is, right? They all seem to share the same properties of an intelligent agent. But what the evolutionists do is say, well, it looks like a duck, it walks like a duck, but it's not, it's a cow. ID proponents say, well, it looks like a duck, walks like a duck, because it's actually a duck. That's the difference between both sides of the scientists at both sides of the fence. And when you say that the vast majority of scientists agree with you, they don't. They would agree with us.
So I'm sorry, that's simply untrue. But beyond that, the biggest issue with the ID proponents is they do not engage in the publication of their ideas and defend them rigorously in the academic sense. They do not seek to publish in scientific journals. They, in fact, tremendously avoid any kind of peer review. Famously, Michael Behe's own biology department broke away from it and wrote a letter in the Dover case about ID in schools in Pennsylvania, saying, while we respect Michael Behe and academic freedom and respect his right to speak his mind, we're not convinced. We don't side with him with regards to his ideas. So essentially,
hold on a second.
The difference is the ID proponents are not engaged in science. They are creating camps where they talk amongst themselves, even making their own.
Getting away from the science.
Even making their own journals.
Okay.
And publishing ID articles in those journals as if they were peer-reviewed science journals. And that's part of the problem. But I want to get back, and not to cut you off, by all means respond as you will, but you seem to not want to address this topic of this, to say, or at least try to address it more directly, that if at best we're saying we have example of undesigned or unintelligent designed turbines because the turbines are being built by these bacterial cells. And you can say, well, they're being built by a pattern that's made by something intelligent. But if you're saying we have another answer to how these codes are made, Richard Dawkins's Methinks It Like a Weasel experiment showed that to be simply incorrect because a program, a computer program, I'm not sure if you're familiar with this. I'll link a link to it just so you can have the sauce for this.
Yeah, I know what you're talking about.
Essentially, a unintelligent program was able to output through random changes in selection alone the Shakespearean phrase, "Methinks it like a weasel." So in the case, the analogy here, which is sometimes missed, is if you come across the words, "Methinks it like a weasel," in some context, that information, is that information the result of an intelligence, someone like Shakespeare, or is it the result of an unintelligent computer program? In the case of that, there's a real serious question because it can be shown, much like my analogy with the antifreeze proteins in Arctic fish, that there are sufficient forces to make these changes. And whether or not you think that any bacterial flagellum were designed and indeed must be designed, the question becomes, is this bacterial flagellum or rotary motor via ATP synthase intelligent design, or is the result of some unguided process that itself may have been the result of intelligent design?
Okay, well, you've been jumping around from topic to topic.
How am I jumping around? This is directly what we're talking about. The ability of evolution to be an alternative, right?
The ability for evolution to be an alternative to intelligent design, right? You got to understand what intelligent design argues.
I think I do.
If it's arguing for specific patterns that are only known to come from intelligent agents, and you bring up different examples that are not dealing with that at all, like a court case that was done in the middle of some small town, which is not the scientific process, which is the middle of some scientific process. I'm sorry, the middle of some small town, which is not the scientific process at all.
But
that doesn't do anything for me.
You realize that.
Velo, hold on, please.
I'm just wondering.
Hold on, please, because you said a lot, and I don't want you to make me miss it.
I think you're wrong here.
Velo, hold on. I know you think I'm wrong, but I also think you're wrong, Velo. Hold on, please stop. The problem is that when we're talking about a specific topic as to whether or not there are certain patterns in nature that are best explained by an intelligent cause, and I get into those specifics, and then you go out on a tangent like you just did, bringing up a court case that was done in the middle of some small town, which is not, court cases are not peer review, right? There's no reason for you to bring that up.
So I'm sorry.
Hold on, Velo, please. Velo, you gotta stop interrupting me, please.
When the time is
Velo, please stop interrupting me. You're gonna make me
a question.
Please. I get that. But please let me finish talking, please. Please let me finish talking, please. So then you start talking about, well, Methinks Like a Weasel, which was actually, there's a lot that goes with that. I won't get into all the specifics of that right now, but we'll get into so we won't get off topic on that. When you're talking about intelligent design, you're talking about certain patterns that have only been known to come from an intelligent agent. If I look at certain, I drop a couple toothpicks on the floor, and they form the word "it." I can grant you that. That can be conceivable as being information. But if I drop a declaration or I drop a semi-truck full of toothpicks and then they form the Declaration of Independence, the specific arrangement of those characters to give overall meaning, that's a different property that has not been accounted for by anyone on the evolutionist side, which is the reason why I brought up the confusion earlier on in this hangout in regards to the information theories and Shannon information. Shannon information, which is typically applied to evolutionary theory, can reduce uncertainty regarding the characters of the information chain, down an information chain, but they don't go into specific sequences like you do with the needed information inside of life in regards to DNA. So you went way off topic. If there is an explanation, an alternative explanation, which can accounts for something like the ATP synthase, something very specific where you can't go off topic and cause confusion, then that's what I would like to get into. And you claimed earlier on that this article that you gave me was evidence for evolutionary theory, accounting for this particular system. Well, I would like for us both to read through it right now so that we're not just, trying to citation bluff and see if there's actually some evidence on your side. So I'm going to read this through. I'm going to read this if I can, so that we can look through it.
So, if I may, I'm not trying to say that you're being disingenuous here, but when you raise the specter of to say, well, ID scientists carry to the logical conclusion: if it walks like a duck, talks like a duck, quacks like a duck, it's a duck. I responded with the reference that no, even the actual scientists, biologists, though there are very few biologists in ID proponents, those that propose these ID hypotheses do not do what scientists do. They do not seek to publish their ideas in the peer review. They do not use the term intelligent design, nor do they defend it to their peers. And my reference was not about the court case. It was about Michael Behe, who testified in the court case, an ID proponent.
Well, you misunderstood what I said then.
The specific citation that I gave was the fact that Michael Behe's department, his own biology department at his university in Pennsylvania, or I think it's wherever it is yet. He may have moved since I last checked, but they themselves wrote a letter, unprompted, saying, we respect Dr. Behe, we respect his independence and his academic freedom.
You're missing the point.
We do not see his views as being valid. So what I was trying to address,
you're confused.
Finish my point as I tried to let you finish. What I was trying to address was the major difference between even the scientist with ID, the scientist with a specific field like Stephen Meyer or William Dembski or Michael Behe or Jonathan Wells. These are people with real science degrees who actually do real science or have done real science. They do not seek to propose, defend, or support their hypotheses in the scientific literature.
That's not true.
That simply is true. That is a
That's
fact.
not true at all. Stephen C. Meyer had an article that was published, and there was a whole investigation that went down because of what happened with Richard Sternberg and so on. That's just not true.
Yes.
He had an article that was published, and there was a whole investigation that proved that the evolutionists were most definitely trying to be. Hold on, Velo, please stop interrupting me, please.
Well, I wasn't.
What you just said was wrong. But what you just said was wrong. There was an investigation that proved that the evolutionists were malicious in regards to an article that was published by Stephen C. Meyer. That's just wrong. It's 100% incorrect. So, and after this hangout is over with, because you've made a bunch of statements here, and I want to try to tackle them as best as I can. But after this hangout is over with, guys, I'll give you guys a link so that guys can go and look into the investigation yourself to show that this is 100% incorrect. But you were confusing what I just said right there. Your response was really incorrect, because the point that I was trying to illustrate here was that when you look at what the scientists say on the evolutionist side and on the intelligent design side say, they are all in agreement with what the data says in regards to what ID proponents are claiming. The ATP synthase is a turbine. The coded information that you're observing inside of the cell do share the exact properties that we know come from an intelligent agent. And so the point that I was trying to help you understand was that the evolutionists are saying, well, it looks like a duck, quacks like a duck, walks like a duck, but it's a cow. And the ID proponents are trying to say, well, it looks like a duck, quacks like a duck because it's a duck. They're coming to the only reasonable conclusion based on the data. And just like when I'm dealing with you, this is what the ID proponents deal with. They're not getting any sort of sufficient answers that you would expect the scientists to give. They're getting off topic and they're not dealing with it.
I'm simply sorry to say that you're incorrect. In the case of the von Sternberg affair, I don't know what conspiratorial argument you'd put forth, but Dr. von Sternberg was in fact an unpaid volunteer, and he circumvented the standard peer review protocol.
That's okay. So what I'm going to do?
I'd like to finish.
Hold on a second.
I would like to finish my statement, true empiricism, and not let you interrupt me yet again.
What I'm going to do is I'm going to
complain.
But you keep Velo, please, Velo. I know. Let me say this one thing. I know.
Are you going to let me finish my point, or are you not going to let me
finish? I'm sorry. I can't because you keep on jumping over this article. You keep on trying to avoid reading this article. You've been claiming that this article proves your position. I'm going to post a link below so that people can read it themselves after this thing is over with. I promise you. They can look into it themselves. I already know about all the claims and accusations that the Smithsonian were positing so that they can try to get themselves out of that mess. But there was an investigation that proved the opposite. So I'll put the links below after this video is over with so that people can look into it themselves rather than, with all due respect, being misled. They can look into the actual investigation themselves so they can know that the complaints of the Smithsonian, it's just bogus. So let's look into this supposed.
Well, I'm sorry, but are you simply going to mute me when you don't want me to make my point?
Well, you keep on jumping over your claim. You keep giving me these articles, but you don't want to read through them.
Finish the point, and we can move on if you want to. But the point I'm trying to make here, beyond just the objections to the improper way in which the article was initially published, in a field that's not really suited for this argument.
Let's read your article.
The issue
let's read your article.
The issue at hand, which I wanted to raise, is even if you read the article that Stephen Meyer,
I'm going to post the link below.
I'd be happy to read that article, or I wish everyone would read the article. It didn't advocate ID. And the objection I'm raising is when you say, oh, there's some packet of information that can be inferred to be designed. There is no, and there never has been, a rigorous definition proposed by the ID proponents, and that's a major problem.
Okay, well, you're incorrect about that because at the bottom of that article that was published, there was a positive case for intelligent design.
Where was the intelligent design?
Like I said, you keep on trying to move away from certain topics. You gave me this article here. I would like to read through this article so that we know that.
Yes, in true empiricism, in the future, I would not now, of course. I would like to discuss the article and specifically have you point out where in the Stephen C. Meyer article you think it's advocating for a positive case for ID.
I have no problem with that at all.
In the future, of course, let us go to the article with which I pointed you to, specifically titled "The Evolution F0 F1 ATP Synthase Complex in Light of Patch Distribution of Different Bioenergetic Pathways Across Prokaryotes." So what's about this doesn't address the question that you wanted. Like, what question you wanted to address, basically. Let's start there.
Okay, so the article is going to be placed below, guys. You guys read through it. I'm going to try to find it before I head to bed tonight. What I want to look through right now is here's the journal the article that he gave me probably over an hour ago. And supposedly this was evidence for the claim of evolution. Here's the abstract. Bacteria and archaea are characterized by amazing metabolic diversity, which allows them to persist in diverse and often extreme habitats. Apart from the oxygenetic photosynthesis and oxidative phosphorylation, well-studied processes from chloroplasts and mitochondrial plants and animals, prokaryotes, utilize various chemo or lithotrophic modes such as photosynthetic. What is it specifically that you think in this article proves your point?
Well, so the setup is to talk about the different energetic pathways, because as hopefully you know from basic biology, the evolution of chemoautotrophs is thought to have occurred first. And when we're talking about ATP synthase, that is something found in non-autotrophs. So basically heterotrophs and the organisms that seek energy from other pathways. So when we're talking about this, we're juxtaposing the notion that there are other energetic pathways that are used to produce ATP. There had to have been, in accordance with evolutionary thinking. And the hypothesis would be, well, we'd say that some of those pathways may have been co-opted through various evolutionary mechanisms into form new structures that could perform what we're seeing today. So it's testing that hypothesis by saying, do we see the data, what we see present in extant organisms, matching the hypothesized expectation?
So basically what it's doing is comparing homology again then.
No, in this case it's saying, do we see that the subunits, the structures of ATP synthase being explainable as being modified from previously?
Yeah, I'm reading through it now. So while each pathway has been studied in considerable detail in isolation, not much is known about their relative evolutionary relationships. Okay, you can't get anyone directed that. Wanting to address how this metabolic diversity evolved, we mapped the distribution of the 9 bioengineered modes of phylogenetic tree based on the 16 sRNA sequences from 272 species representing the full diversity of prokaryotic lineages. This highlights the patchy distribution. This is in the first paragraph of the article. The first paragraph of the article, it says exactly what I said the problem is with evolution.
Wait a minute.
Wanting to address how this metabolic diversity evolved.
Why are you scoffing at that? Why is it something to scoff at that scientists say we don't fully understand these relationships? Let's do an experiment.
Okay, so let's keep reading. Wanting to address how this metabolic diversity evolved, we mapped the distribution of the 9 bioenergetic modes of phylogenetic tree based on the 16 sRNA sequences from 272 species representing the full diversity of prokaryotic lineages. That highlights the patchy distribution of the many pathways across different lineages and suggests either up to 26 independent origins of 17 horizontal gene transfer events. Next, we use comparative genomics phylogenetic analysis of all subunits of the F0, F1 ATP synthase common to most bacterial lineages regardless of their I'm sorry, guys, it's late. Energetic.
Well, can I help you? Why don't you skip to the next paragraph, which is the author.
Indicates an ancient origin of this protein complex and no clustering based on the bioenergetic mode, which an ancient origin of the protein complex and no clustering based on the bioenergetic mode, which suggests that no special modifications are needed for the ATP synthase to work. The different electron transport chains, moreover, explanation of the ATP synthase genetic locus indicates various gene rearrangements in the different bacterial lineages, ancient duplications of the ATPL. I'm sorry, guys, I'm scrolling down.
Yeah. And like I said, this might help if you just go to the author's summary. But we can get to that too if you want to.
All right, I'll read through the whole thing. I don't have a problem with it. I'm up as it is, so why not?
Well, it's just, to me, is this just homework time? Is it time for you to, I'd prefer you actually read this article and discuss it in the future, because it seems that this is the first time you're seeing this. And so
I've read through quite a bit of this, but
at this point in time, how serious I should take you if you're saying there's no other viable explanations when you're not even familiar with some of the standard publications in the field regarding the evolutionary model with how these things evolve.
Well, I have. That's why I brought up the hexamerichelin cases earlier. There's a lot of different examples that we can use to try to. I mean, there are people who believe that the ATP synthase that evolved inside of the. I can't hear myself talk when.
Yeah, this is from 2014. You haven't read it yet?
Yeah, I think that I have read this, but I want to read through it again. I mean, I haven't been online in a long time looking through this stuff with people, man. So I want to look through it again and make sure that it's going to say what you're saying. So bacteria and archaea are the most primitive forms of life on Earth, invisible to the naked eye and not extremely varied or impressive in their appearance. Nevertheless, they are characterized by an amazing metabolic diversity, especially in the different processes that use to generate energy in the form of ATP. This allows them to persist in the diverse and often extreme habitats, wanting to address how the metabolic diversity evolved. We map the distribution of the 9 bioenergetic modes across all the major lineages of bacteria and archaea. We find a patchy distribution of different pathways and suggest either frequent innovations or gene transfer between unrelated species. We also examine the F-type ATP synthase, a protein complex which is central to all bioenergetic processes and common to most types of bacteria regardless of how they harness energy from their environment. Our results indicate an ancient origin for the protein complex and suggest that different species, without necessitating major innovation, use the pre-existing ATP synthase and adapt it to work with different bioenergetic pathways. We also describe gene duplications or arrangement of ATP synthase subunits in different lineages, which suggest further flexibility and robustness in their control of the ATP synthase. So basically what's going on is exactly what I said earlier on. They were comparing different lineages of the ATP and different ATP lineages, and they were assuming common ancestry.
Well, but they're not assuming.
There's a graph right over here below.
They're using a phylogenetic analysis to show the interrelation between different types of this present in the bacteria.
Oh, I can't hear you. Hold on a second.
But I just want to be clear. What they're saying is they're looking at the extant bioenergetic processes of this system in different extant species. In the case of different bacteria, with regards to how they essentially consume energy, how they gather energy from their environment, and they're showing that the most likely explanation with regards to these mechanistic pathways is, as I said, not major innovations. That would be minor or non-major changes to previously existing structures.
Yeah, that's what I just read.
Yes, I agree. So then that's the evolutionary.
What did I tell you earlier?
Is it homologies? Is it gene duplication? Is it a mechanistic explanation?
Basically, here's what's happening. Okay, they realize that there are certain changes in these different structures of the types of ATPs that there are.
There's diversity.
There's diversity, right? There are diversity. So what they're basically trying to say here is that those changes that are needed, this is likely the pathway, that the direction that we went, right? The direction that we need to go in order to show the lineage here with the ATP synthase. Or
what changes need happen to have what we have today? Yeah.
Yeah, it's telling me what needs to be changed so that you can build this structure. It isn't giving me an answer as to whether or not it's actually true that the structure can be changed.
Well, wait a minute here, because that's a bit of shift in the goalpost. Because what we're saying here is we're describing what we can hypothesize about these interrelations in terms of their origins.
No, we're not.
And we're saying, how can we test this? And we test it by looking at the diversity within living organisms.
So
seeing homologies.
So let's
seeing the genetic basis, the gene duplications, the mutations,
and
how even there's horizontal gene transfer among different lineages.
Listen.
So to reiterate the point that they made, I think this deserves to be repeated here, is they say our results indicate a ancient origin for this protein complex and suggest that different species, without necessitating major innovations, used their pre-existing ATP synthase and adapted it to work with different bioenergetic pathways. And that's, I think, the point of evolution, because it's meant to describe an explanation for the origin of diversity. And it does so by saying what previously existed was adapted to suit new purposes. At what point are you disagreeing with at least the efficacy of evolution to act as this explanation with regards to ID?
Because it's very simple, okay? When I was arguing with you earlier on, I said that there are certain explanations that can only account for the complexity of the ATP synthase, because based on those patterns, we observed that coming from an intelligent agent. when we were talking about evolutionary theory, I made a very careful point to point out that we can look at the diversity of organisms in the fossil record, and we can assume one organism probably diverged from another organism because of their similarities in stature. That doesn't tell me, because somebody comes with a preconceived notion that that we all share a common ancestor, that they therefore share a common ancestor because of their similarities. And I use an example of a skateboard and a motorcycle, for example. So here you're giving me an article to explain the lineages and the needed differences between those lineages, the differences in those lineages, which would suggest common ancestry. That's not
what it's saying at all. I'm sorry, you missed the point of the article then. This is just the abstract, not even the article.
What did I miss then? What are you talking about?
The problem here, and you've kind of raised it, is to say you keep on asserting that somehow design is in fact, the only explanation we have access to. And I think that's the issue when we talk about explaining diversity. Because when we're dealing with the notion of...
See, it says that bacteria and archaea are the most primitive forms of life, invisible to the naked eye, and are not extremely varied by impressive of their appearance. Nevertheless, they are characterized by an amazing metabolic diversity, especially in the different processes they use to generate energy in their form of ATP. This allows them to persist in diverse and often extreme habitats. Wanting to address how this metabolic diversity evolved, they mapped the distribution of the 9 bioenergetic modes across all the major lineages of bacteria and archaea. So they look at all the major lineages between the bacteria and archaea. We find a patchy distribution of the different pathways. They look at the patchy distribution of these different pathways, which suggests either frequent innovations. They're just pointing out that there had been several changes, or gene transfer between the unrelated species. We also examined the ATP synthase, a protein complex which is essential to all biogenetic processes and common to the most types of the bacteria, regardless of how they harness energy from the environment. So just like I'm trying to tell you now, imagine if you have a book, right? This is an example that I typically use here on my channel. If you have a library full of books, and imagine that there was no author written on any of those books, but all of those books were utilizing the English alphabet, right? You can take one book off the shelf and compare it to a smaller book, and you could probably find similarities in sequences and how many times the or different words are used, and compare them to each other and say, well, these are similar to one another. These have some of the same words and paragraphs. It's the exact same thing here. This doesn't therefore mean common ancestry, because you point out, well, these are the differences that you see in this lineage. Here's the differences that you see in these lineages. This is what you're going to need to change to be, obviously over here, these are the changes that happen over here. But it doesn't tell me anything, just because you can point out, here are the changes over here. This is how different this organism is over here, or this lineage is over here compared to this lineage. It doesn't tell me anything. And so likewise, with the book analogy, just because you might find similar sequences and how those characters are used in each one of those books, it doesn't mean, therefore, a common author. And likewise, with these different lineages inside of these I'm sorry, you guys. It is really, really late over here. But
I was actually trying to make a point.
These different lineages. I just read the author summary again, and these different lineages that you see in bacteria and archaea. It's just pointing out what I've been bringing up in this room this whole time. The article doesn't.
So the point I was trying to raise here is.
Give me a second. I'm going to be right back.
Right? Yes. Okay. Well, I'm not sure I should continue since he stepped away, but the point I'm trying to raise, hopefully True and Pierce can still hear me. If not, I'll repeat myself. Is what's put forth is somehow the divine inference has explanatory power, and I don't really see that as being demonstrated beyond just this.
Hold on, I've been having a hard time hearing you. I need to put my headphones.
Sure, that's cool. No problem. I can wait. Tell me when you're good to go.
All right, I got you. Go ahead.
All right, cool, man. So the problem we have here is to say, on one hand, we're trying to examine a known, established explanation that has been studied for hundreds of years and in various degrees of rigor and put forth and examined in the peer review with scholars of all different backgrounds and views. In the case of ID, we have a group of individuals who may or may not be tied strongly with religious convictions. That's a separate issue, of course, who do not seek to publish their views widely. Let's just go with that. In scientific literature. And the issue we have here with your explanation is to say, in the case of explanation, with the case of the ATP synthase alone, what's being put forth here is the idea that it was modified from previously existing structures. Because evolution says we have diversity. We seek to explain that diversity through unintentional, unminded, non-intelligent mechanisms which modify previously existing structures based upon different selection inputs. So in the case of that, we'd say, well, we expect to have certain homologies. We expect to have previously existing things being modified in a fairly stepwise fashion. And that's the case of what this article seeks to describe. And the problem we have here with the case of the intelligent design hypothesis is to say, why would it be the case?
Well, I think you're missing.
Wait a minute, if I may, this is the point. Would a designer need to modify things in a stepwise fashion? Why aren't there completely distinct lineages of the ATP synthase? Why aren't they completely disanalogous from the notion of horizontal gene transfer? Would it also would the design hypothesis work equally well in the case of a stepwise fashion of things being changed, the Tinkerer hypothesis, as opposed to the de novo creation of independent, unrelated ATP synthases, different designs of rotors with no clear analogy in terms of a stepwise modification?
Do you notice something? The way that our discussion first began was that I pointed out the main issue that I have with evolution, and it's that there are no articles out there that exist that can point out that the accumulation of changes in the genome can accumulate to build or account for a major anatomical feature that we observe, and specifically said above the genus. And then I went on to say that there are certain examples, like the ATP synthase, where there are no tests in the laboratory in regards to the mechanisms of evolution that can account for these particular types of systems. And all that I have here with this article that you gave me, this is the article that you've given me, is pointing out lineages, pointing out those needed changes within those lineages, and then declaring common ancestry because of the similarities and differences that you find within those lineages. And likewise, like I said inside of the book analogy, you can throw a couple books on the table and you can line up their sequences and how many times the English language is used, and how many times those different words are used in those books, and say, well, this one is more so related to this one, or this one's more so related to this one, in the sense to say, well, there's a common author because you find a similar way in being written or whatever. But it doesn't answer the question. You don't go nowhere. You don't get anywhere by declaring common ancestry because you can perceive in your head the necessary patterns that are going to be needed for them to be related.
I just
the point is that I'm saying I know what would be needed for these different lineages to occur. It makes sense for me to say, well, an intelligent agent can arrange these components in such a way to give these different structures their function. Well, you say, well, no, it was evolutionary theory. You can see it by using your imagination. And
so I'm sorry. Did you actually hear my objection that I put to you just now?
What's that?
So I mean, again, this may be a mechanism issue, but that's the point I raised. Is one, that's not what this paper seeks to do. It's specifically pointing out this pathway. It's suggestion. It's concluding.
Why did you give me it?
It's not just the matter that they look the same. It's saying that they were modified in a way that did not require major innovations, and even tracking.
So they trace. Okay. Hold on. Wait, right there. Wait. That's important right there. So they observe the modification now. That's not what I just read. What I just read was they traced the. Okay, it's like if I put a couple books on the table, I can point out these words have to be arranged this way for it to be closer related in sequences to this book right here. That's obvious. Anybody can point out the obvious. This isn't like this book because of this reason. It's different because of this reason, right? And in the same way, if I look at those lineages in that article that you gave me and the exact portion that you told me to read, that's not going to get me anywhere, right? Because if they're just telling me, okay, well, this one's different from this one, and this one's different from that one, so they're closely related. I mean, it's not getting anywhere.
Yeah, I agree.
I'm tired.
You actually have to read the article.
I'm very tired. I'm very tired.
What I told you,
I don't think you read it. With all due respect, I don't think you read it.
But I'd like to finish my point without you interrupting me, because that's kind of the point.
I got to get to bed, man. I've been doing this way longer than
I will happily rest on the point that you seem to be having trouble addressing, which is at the core why the ID argument fails. Is because it is at some level unfalsifiable, because of the notion of what would be consistent.
It's not unfalsifiable.
If I may, my point is, what I'm saying is, what would be consistent with design hypothesis? Now, in the case of evolution, as I said before, it proposes to manifest these eventual outcomes, the case of the phenotypes of living organisms, by modification of previously existing structures. And the article, that I referenced,
you don't ever get to test those mechanisms being modified. That's the problem.
To address this, going down to the individual genes associated with subunits as they relate to different bioenergetic pathways. So we combine that with my objection. My challenge to you is to say, what is consistent, or more so, what is inconsistent with the design hypothesis? Because in the case of evolution, we would require a relatively stepwise, relatively simple modification that could be selected for. With regards to the design option, could the designer have done so, the tinkerer designer, making small modifications along the way? Or if we saw completely unrelated structures, if we saw the rotors, as you say, the turbines, ATP synthesis with no clear analog that seemed to develop with completely different structures, although accomplishing a similar function, independent in terms of their genetics or in terms of their actual morphology, the phenotype of the actual protein. In both cases, would those be consistent with the design inference?
So you've not listened to anything I've said.
Could you respond to that point before we move on?
So
before you leave,
let me try to say it one more time, because I don't know if you're listening. I don't know if you're listening to me. I don't think you are.
I'm trying. I'm not hearing your response to this.
Can you stop? I'm in the middle of speaking. Can you stop?
Well, I actually want you to respond to the question, please.
Okay, so I'm going to respond to you now, but I'm going to have to do this. I'm sorry. So here's what my point was. Okay, I pointed out earlier on in this hangout that just by pointing out similarities in stature or similarity in already existing features, it doesn't prove the point that they therefore evolved from a common ancestor. The idea that something can evolve and share relation is a presupposition that first you need facts on the table before we can believe that. If you come into this argument believing evolutionary theory is true, then you can go ahead and say all you want by pointing out the similarities in already existing features that they all share a common ancestor by arranging the least similar in one direction and the most similar in another direction, put them in this tree and assume they're common ancestor. You can do that all day long. You have that belief, that's fine. But it's a religious belief. It's not a scientific belief. When you start saying that these similarities, like you're pointing inside of these lineages, have been traced, and then those similarities can be observed, now you're saying something else. That article that you just gave me right now, it points out the lineages, and then it points out those differences in those lineages, and there's no problem there, okay? But what it's doing is it's superimposing upon the differences and similarities of those lineages and assuming common ancestry. But it's not answering the question. It's staying within the confines of the religious belief, but not answering the question that I asked you originally in this hangout. Originally in this hangout, I asked you, whether or not there have been tested mechanisms in the laboratory to show that these events can actually occur. That's the point here. If there is actually some sort of evidence out there that can show that the mechanisms of evolution have been tested to account for these particular systems being developed, then the burden of proof is on you to show that. Thus far, you haven't given me that. You've given me, and I read your portion of your article that you gave me, and you claim that you read it. I don't know if you read it or not. I don't think you read it. When I read through that article, it didn't say what you bluffed me on earlier on in this hangout. You used it as an explanation to account for the diversity the mechanisms of evolution accounting for these particular systems. It didn't show that at all. It showed the original point that I was trying to prove to you earlier on. So that's the point here. I don't care about a religious belief. I don't care about you superimposing on information and saying, well, look, you can trace the similarities. I get all that. I get that you believe that if you allow small changes to occur over time, then you can share that some common ancestry is available. I get you believe that. I get the fossil record. I get phylogeny. I get what you believe when you picture in your head. But the point is that if this is a scientific theory, then there needs to be a tested mechanism in the laboratory to see whether or not this change can actually occur. Earlier on in this hangout, I even used another analogy of the gumballs, for example. You have 3 cups on the table, cup 1, cup 2, cup 3. And you have a bag of gumballs in your hand. Each gumball represents change over time, right? And you throw the cups in the gumball in cup 1, they accumulate, they pile over, they don't do anything. You throw the gumballs in cup 2, they rip the cup open and they flow out from the bottom. In cup 3, they arrange inside of the cup and they build distinctive shapes, right? You're saying that but by observing the already existing lineages, you using your imagination and connecting the dots, then therefore this shows common ancestry, while never testing the mechanisms to see whether or not your conclusion is true at first. We don't even know if that's a possibility yet. These are already existing features. We don't know what the conclusion is if you allow the change to happen. You can speculate, but we don't know what the conclusion is. We've never had a test to allow changes to continue to happen to see what the end result is going to be. When we look at the 4,400 generations of pathogens, it seems like the organisms, they deplete in stature. When we look at the fruit fly experiments that have been done, and they've been doing experiments on them for the last 100 years, and now increasing the mutation rate by 1,500 times using X-ray machines, we see that the organisms tend to deplete in stature. That they have different reasons for utilizing these fruit fly experiments, but it's like every experiment doesn't show when we're putting it to the test the result of what you're claiming. So we don't even know whether or not the accumulation of changes can build any of these features. We don't know if that creative ability is actually available. You've given me an article that just repeated the problem that I pointed out several times in this room already, and you actually used it as a bluff before we read into it, and then after we read into it, it proved my original point. So I'm going to end the hangout now. Your volume is increased again. But maybe tomorrow, maybe we could set up a debate or something.
No, sure, yeah. I hope to talk again. It just seems that you're not really addressing, like you did not answer the question I put to you, nor the challenge I raised with regards to the efficacy of ID. I don't know if that's something you should think about. Like, I suggest you just think about that in the future. Because I really don't know, just beyond any particular conversation, you seem to be bouncing back and forth between the notion of, do we have an explanation for this specific feature, which is not anatomical, I'd say. It's part of the cellular mechanism. But combined with, does evolution have the efficacy to enact such?
Guys, I really, you got to end this.
I just want to emphasize, I tried to address that with regards to the evolution of the antifreeze proteins in Arctic fish, because we have shown a stepwise fashion for that. So in the future, hopefully we can discuss this more.
Okay, sure. So that's it. Okay. If you want to have another discussion, I'm not trying to be rude. I know I've been
really patient. Please.
Well, I slept 4 hours yesterday. That's why, and I got to be up early tomorrow. So
no problem.
If you want to have a talk about this sometime in the future, I'm not trying to be rude. I'm really tired, and I
think you
just
were
got a call. I got a call just now, so I got to step out too.
Do what you got to do, man. We'll talk to you next time.
All right. Take it easy.
Be safe out there.
All right.